Welcome to the Concierge Care Health System podcast. The information shared in this podcast is for general informational purposes only and does not constitute medical advice. Listeners should be aware the content is not a substitute for professional medical advice, diagnosis, or treatment. Always consult with a physician or other qualified healthcare provider for medical questions.
Welcome to the deep dive. You've definitely heard about dopamine, right? It gets tossed around a lot as the brain's pleasure molecule, the thing behind feeling good.
Yeah, that's the popular idea.
But what if I told you that's really just, scratching the surface? Dopamine is actually fundamental for so much more. It helps control how you move, it fuels your motivation, plays a huge role in executive functions like attention and planning.
Absolutely.
And here's where it gets, for me, really fascinating. What if this crucial system isn't the same for everyone? What if it's fundamentally different depending on whether you're male or female?
It's such a critical question because, honestly, for way too long, neuroscience research basically treated male and female brains as, pretty much interchangeable.
Right, like a default male brain was the standard.
Kind of, yeah. But now we have this burgeoning body of evidence, really strong stuff showing profound, functionally significant differences in the dopamine system based purely on sex.
So our mission today is to really unpack these distinctions. We're going from the tiny molecular level all the way up to how this impacts health and behavior in the real world.
Exactly. It's a deep dive into why being male or female genuinely changes how this vital system operates.
And look, understanding these sex differences isn't just some academic exercise. It's actually critical for public health. For your health, really. Think about it. Conditions like Parkinson, schizophrenia, ADHD, major depression, they all involve dopamine somehow going wrong.
Right. Dopamine dysfunction is key.
And they all show up differently in men and women. If we ignore these differences, we risk, misdiagnosing things or developing treatments that just don't work as well for half the population.
Which has definitely happened.
The good news, though, is that major institutions, like the NIH in the U.S., they now formally require researchers to consider sex as a biological variable.
Yeah.
So the importance is being recognized.
Which is a huge step forward. So to help everyone listening grasp this, it's complex but also fascinating. We'll start by laying out the basics. How dopamine works, how it's made, travels, sends signals.
The groundwork.
Exactly. Then we'll dive into how hormones, specifically estrogen and testosterone, really sculpt its functions so differently in males and females.
Okay.
And finally, we'll connect all that to specific ways these differences actually show up in brain function and behavior, and maybe most importantly, the clinical implications. What does it mean for disease and treatment?
All right, let's get into it. To really appreciate how these sex differences work, we first need that basic picture of dopamine's architecture. So how does the brain actually make this stuff and move it around?
Okay, so think of dopamine like a chemical messenger. It's built from simple stuff you get from your diet, amino acids.
Right.
Your brain has this specialized assembly line with key workers. These are enzymes that turn these building blocks into dopamine. The main one to know is tyrosine hydroxylase, or TH. That's the bottleneck, the rate-limiting step.
Got it. Sets the pace.
Exactly. Once it's made, it gets quickly packaged into these tiny delivery trucks, vesicles, inside the neuron by another protein called VMAT2.
Okay, packaged up, ready to go.
Yep. Then when a signal comes along, these trucks release the dopamine into the synapse, that tiny gap between neurons, so it can deliver its message.
So it delivers the message, but then what? It can't just hang out there forever, can it? That would cause chaos.
No, absolutely not. Its action is really brief, very tightly controlled. There are specialized cleanup crews, other proteins. The main one is the dopamine transporter, or DTA. DTA quickly scoops the dopamine back up into the neuron it came from, recycling it. Or other enzymes like MAO and COMT break it down into inactive stuff like HVA.
So it gets cleared up fast.
Super fast. That cleanup is crucial for the system to stay precise and ready for the next signal.
Okay, so that's how it's made, managed, cleaned up. But dopamine isn't just, floating around randomly. It travels along specific routes, these sort of neural highways. What are the big four pathways we need to know about today?
Right. There are four main ones. First, the mesolimbic pathway. This one runs from a deep brain area, the ventral tegmental area, or VTA.
VTA, hear that one a lot.
To the nucleus accumbens, or NADOC. This is the famous reward pathway. It's central to motivation, that wanting feeling, incentive salience, and positive reinforcement.
The wanting system. Makes sense.
And when this pathway gets overactive, that's strongly linked to things like addiction and also the positive symptoms of schizophrenia, like hallucinations or delusions.
Okay, so VTA to NAC is reward and wanting. What's next?
Next is the mesocortical pathway. This one also starts in the VTA, but it projects to your prefrontal cortex, the PFC, your brain's CEO, basically.
The thinking part.
Exactly. This pathway is crucial for higher order stuff, executive functions, things like working memory, attention, planning, impulse control.
Right.
Now, if this pathway has too little activity, hypoactivity, that's linked to problems like the negative symptoms and cognitive issues in schizophrenia, things like lack of motivation, anhedonia, trouble concentrating.
Okay, so we've got reward motivation and then higher level thinking. You mentioned 4 pathways. What's the biggest one?
That would definitely be the nigrostriatal pathway. It's the largest by far, carries something like 80% of all the dopamine in your brain.
Wow, 80%.
Yeah. It runs from a different area, the substantia nigra pars compacta, or SNC,
Okay.
to the dorsal striatum, which includes the caudate and putamen. This pathway is the main regulator of voluntary movement, motor planning, learning physical skills like habits.
So movement control. That sounds like Parkinson's.
Exactly.
Yeah.
The loss of dopamine neurons right there in the SNC feeding this pathway is the absolute hallmark of Parkinson's disease. That's what causes the tremors, rigidity, the movement problem.
A massive pathway.
Yeah.
So that's 3. What's the 4th?
The last one is the tuberoinfundibular pathway. It's a shorter circuit running from the hypothalamus to the pituitary gland.
Okay. Different function.
Yeah, it's mainly neuroendocrine stuff. Its main job is inhibiting the release of prolactin, a hormone involved in milk production. Okay, so that explains why some drugs, like antipsychotics that block dopamine, can sometimes cause high prolactin levels.
Precisely. Blocking dopamine here takes the brakes off prolactin release.
Fascinating. So dopamine gets made, packaged, sent down these highways, but how does it actually work when it arrives? It needs to bind to something, right? The receptors, like a lock and key.
Exactly like a lock and key. Dopamine exerts its effects by binding to specific receptor proteins on the receiving neuron. There are 5 main types, D1 through D5.
5 types.
Yep, and they're usually grouped into 2 families. The first is the D1-like family. That's D1 and D5. Generally, these are excitatory. They tend to activate the neuron, ramp things up. They're involved in working memory, attention, movement control.
Okay, D1 like revs things up. What about the other family?
That's the D2-like family, D2, D3, and D4. In contrast, these are generally inhibitory. They tend to decrease neuron activity, dial things down.
So D2 like puts the brakes on? Pretty much. And the D2 receptor itself is particularly crucial. It exists in different forms, and it's the main target for almost all antipsychotic medications and also drugs used to treat Parkinson's. So understanding these locks is absolutely key.
That's a brilliant overview of the basic mechanics. Okay, now for the real deep dive part. Sex differences. How do hormones like estrogen and testosterone come in and shape this whole system differently?
This is where it gets incredibly compelling. Let's start with estrogen, particularly the main form, 17-estradiol or E2. In the female brain, estrogen often acts like a master conductor for the dopamine system.
A conductor, how so?
It influences dopamine at so many levels. It can bind to receptors inside cells, nuclear receptors, and these then regulate gene expression, basically telling the cell to make more or less of the proteins involved in dopamine synthesis, transport, reception.
So it changes the fundamental machinery.
Yes, but that's often slower. It also acts really fast through receptors on the cell membrane, quickly modulating how neurons fire and release dopamine. It's got both long-term and short-term effects.
So it's working on multiple timescales.
Yeah.
What's the overall effect? Does it boost dopamine or tamp it down?
Generally, estrogen enhances dopaminergic activity. Consistently, studies show it increases dopamine production, it boosts release into the synapse, and it speeds up turnover, how quickly it's used and replenished. It literally makes dopamine neurons fire more frequently.
Wow.
And here's a key part. This leads to cyclical dynamics. Dopamine activity in women actually fluctuates significantly across the menstrual cycle, generally peaking when estrogen is high.
Ah, okay. That makes intuitive sense.
Yeah.
People report mood changes, cognitive shifts.
Exactly. It has real impacts on mood, cognition, things like working memory can fluctuate, and even things like craving for substances, for instance.
It's a very dynamic system in females tied to these hormonal cycles.
Very dynamic. And estrogen also coordinates things. It upregulates that DEET transporter, the cleanup crew, suggesting the system is set up to handle these periods of high dopamine release efficiently. It fine-tunes the whole orchestra, volume, and tempo. It creates a system characterized by dynamism and plasticity.
That's a great way to put it. Okay, so estrogen as the dynamic conductor. Now let's switch gears. Testosterone. How does the primary male sex hormone shape the dopamine system? Is it doing something similar?
It's quite distinct, actually. Testosterone's influence is both organizational, meaning it shapes the brain's development, especially crucially during adolescence,
Setting things up early on.
and activational, meaning it continues to modulate how the system functions in adulthood. It mainly works through the androgen receptor, AR, but interestingly, testosterone can also be converted into estradiol in the brain by an enzyme called aromatase.
Wait, so the male brain makes its own estrogen from testosterone?
It does in certain areas, so testosterone can exude some effects indirectly via estrogen receptors too. It's complex.
Okay, so what's its main job, especially during that organizational phase in adolescence?
During adolescence, testosterone and its metabolite DHT really help tool up the system, particularly that big nigrostriatal pathway for movement. They increase the expression of genes for the transporter and VMAT2, the packager.
So it's like building a bigger, more robust pipeline.
Exactly, preparing it for high-capacity signaling. Testosterone also fine-tunes the receptors in that pathway, increasing the inhibitory D2 receptors while decreasing others, kind of optimizing the output signal.
So building for high stable performance, how does this connect to typical male-associated behaviors?
Well, testosterone is strongly linked to behaviors involving high motivation, competition, seeking rewards, especially effortful ones. It actually increases dopamine release in the nucleus accumbens, the reward hub, in response to reward.
Fueling that drive.
Potentially, yes. But what's also interesting is that while it builds this high-capacity system, testosterone can also help attenuate or buffer against excessive dopamine activity, particularly in that nigrostriatal pathway. It helps maintain stability, homeostasis.
So maybe less fluctuation than the female system.
That seems to be the case. So if estrogen is the dynamic conductor creating a plastic, responsive system, testosterone is maybe more like a meticulous engineer, building a robust, high-capacity pipeline designed for stable, sustained output.
That engineer versus conductor analogy really helps visualize the difference.
Yeah.
So the core takeaway here is female system, dynamic, cyclical, plastic, influenced heavily by estrogen. Male system, built for high capacity, stable output, shaped significantly by testosterone.
That's the fundamental difference, and it's profound. It sets the stage for real, tangible differences in how the brain is wired at baseline and how we process things like rewards, risks, and how we behave.
So let's get into that. These hormonal differences aren't just acting on identical baseline systems, right? You're saying the wiring itself is different even before adult hormones fully kick in.
That's right. The starting points are different. Anatomically, studies, often in rodents, but with some human correlates, show males tend to have more dopamine neurons in the substantia nigra, that motor control area. Females, conversely, often show more dopamine neurons in the VTA, the origin for reward and cognition pathways. And those VTA neurons in females can be larger and more numerous.
So right from the start, there's a potential bias towards motor control in males and reward cognition in females, structurally.
It suggests a different foundational setup, yes. And we see differences at the molecular level, too. Female rats, for example, show enhanced function of VMAT2, that packaging protein we mentioned, more efficient dopamine handling.
And how does this translate to baseline activity in humans? Can we see this with brain imaging?
We can get glimpses. Human imaging studies suggest healthy women tend to have higher availability or binding potential for those D2-like receptors, especially outside the striatum, in areas like the prefrontal cortex involved in cognition and emotion.
So more listening posts, maybe.
Potentially. Men, on the other hand, when challenged, say with a dose of amphetamine, which releases dopamine, tend to show a markedly greater magnitude of dopamine release, especially in the ventral striatum, that core reward hub.
So women maybe higher baseline receptor availability, men greater capacity for stimulus-induced release. Two different default settings.
That seems to be the emerging picture, and this leads us into reward processing and motivation, where things get really interesting, almost paradoxical.
Okay, the paradox. If men show this bigger dopamine release in response to rewards, you'd expect them to show a bigger behavioral response too, right?
That's what you'd think, and in some ways, it holds true. Human studies do show men often exhibit significantly greater neural activation across the whole reward network, VTA, NAC, other areas when they see reward cues.
So their brains light up more.
Yes, and this often correlates with better accuracy on reward tasks, higher subjective arousal, stronger physiological responses. The male system seems very amplitude-driven, big signal in, big response out.
Okay, that fits the dopamine release findings.
Yeah.
But where's the paradox then?
The paradox comes from animal studies, particularly with conditioned responses. Female rats will often show more robust condition responding. They'll work harder, approach the reward cue more vigorously.
So more motivated behaviorally?
Exactly. And here's the kicker. They do this despite showing a significantly smaller reward-evoked dopamine response in the nucleus accumbens compared to males.
Wait, how do you get more behavior with less of a dopamine surge in the main reward area? That doesn't make sense based on the standard model.
It doesn't, and this baffled researchers for a while. The resolution is absolutely fascinating. It turns out the way the brain strengthens connections to learn about reward synaptic plasticity, or LTP, works differently.
Different mechanisms.
Entirely different in this context. Males use the standard mechanism involving NMDA receptors, the one you read about in most textbooks. Females, however, use a distinct NMDA-independent mechanism. It requires different players. Bell-type voltage-gated calcium channels, and crucially, estrogen receptor alpha, ERI.
So women have a completely separate estrogen-dependent pathway for learning about rewards.
It seems so. An alternative estrogen-sensitive pathway that allows for robust behavioral learning and output, but without needing that massive dopamine surge males exhibit.
That's huge.
Yeah.
That's not just a tweak. That's like a whole different biological strategy for achieving the same goal.
It's incredibly significant. It underscores that we just cannot assume the mechanisms driving motivation, learning, or even disease states are identical across the sexes. And this extends to other areas too, like how we make decisions involving risk.
Ah, risk-taking.
Yeah.
How do the D2 receptors, which you said are inhibitory, play a role here differently?
Well, a key area for weighing risks and rewards is the basolateral amygdala, the BLA. And the function of D2 receptors there seems strikingly sex-dependent.
Okay, how so?
Studies show females exhibit much higher sensitivity to D2 receptor modulation in the BLA. If you activate these D2 receptors pharmacologically, it causes a significant decrease in risky choices in females across all doses tested.
So activating those inhibitory receptors makes females play it safer.
Yes, it seems to enhance their sensitivity to potential negative outcomes like punishment, leading them to choose safer options. Males, interestingly, only show this risk-averse effect at the highest dose of the drug.
But is it always about punishment? What if risk is just, say, potentially missing out on a reward?
That's a great nuance. If the risk is simply the omission of an expected reward rather than an active punishment, some studies show female rats actually exhibit a higher preference for the risky option than males, so it's context dependent.
Wow. So, summing this part up, the male system seems amplitude-driven, bigger dopamine release, more widespread neural activation. The female system is perhaps more efficiency-driven, lower dopamine release for certain tasks, but enhanced receptor sensitivity, and these unique estrogen-dependent pathways compensate to drive robust behavior.
That's a really good working framework, and it provides a crucial lens for understanding how these differences manifest in various clinical conditions involving dopamine.
Absolutely. Let's pivot to those clinical implications. Parkinson's disease, you mentioned it, involves the nigrostriatal pathway. How do these sex differences clearly play out there?
Parkinson's is a stark example. It's significantly more common in men, about 1.5 to 2 times higher prevalence, and women who do develop it tend to get it later in life.
Later onset. Is that linked to hormones?
Very likely. There's strong evidence for a neuroprotective effect of estrogen. It seems women have a higher baseline density of that DTA transporter in the striatum, and maybe more dopamine neurons to begin with, giving them a sort of higher dopaminergic reserve.
Like a bigger buffer against the neuronal loss.
Exactly. It can delay the onset of symptoms. But, crucially, this protection seems to wane significantly after menopause when estrogen levels drop sharply.
So the protection isn't permanent?
No. And interestingly, while men might present with more severe motor symptoms initially, women with Parkinson's, once diagnosed, are often more prone to developing levodopa-induced dyskinesias, those involuntary movements caused by the main treatment.
Ah, a treatment response difference.
Yes. And they also report more non-motor symptoms like depression and anxiety compared to men with PD.
Okay, clear differences in Parkinson's. What about schizophrenia? Another condition heavily implicating dopamine.
Schizophrenia also shows very clear sex differences. Again, incidence is higher in men, about 1.4 to 1, and the onset pattern is distinct. Men typically develop it earlier, late teens or early 20s.
Right.
Women tend to have a later onset, late 20s, early 30s, and interestingly, there's often a second, smaller peak in incidence for women around the time of menopause.
That second peak around menopause strongly suggests a hormonal link,
Right.
losing estrogen's protection.
That's the leading hypothesis. Estrogen is thought to buffer against some of the dopamine system dysregulation implicated in schizophrenia, perhaps dampening excessive dopamine turnover. When estrogen drops, that buffer might be lost for some vulnerable individuals.
And do the symptoms differ between men and women?
Yes, quite significantly sometimes. Men more often exhibit those prominent and enduring negative symptoms, things like lack of motivation, flat affects, social withdrawal, and often have poorer social functioning even before the illness starts. Women are more likely to have affective symptoms co-occurring, like depression and anxiety, alongside the psychosis. And linking back to hormones, some research suggests lower testosterone levels in men with schizophrenia are associated with more severe negative symptoms, potentially related to that mesocortical pathway having too little dopamine activity.
Really striking how these hormonal factors weave through. Let's talk about substance use disorders, SUDs. Addiction is heavily tied to the dopamine reward pathway.
Absolutely. Historically, men have had higher overall rates of SUDs. However, a concerning pattern observed in women is called telescoping.
Telescoping, what's that?
It means that women who start using substances often progress more rapidly from initial use to dependence. They escalate their use faster, tend to experience more severe withdrawal symptoms, and may be more susceptible to relapse triggers.
So a faster, maybe more severe trajectory for women once use begins.
That's what telescoping suggests. And estradiol, that main estrogen, seems to play a key role here. Animal studies show it enhances motivation to seek drugs and speeds up the acquisition of drug-taking habits.
How does it do that?
One proposed mechanism is that estradiol might preferentially boost dopamine release in the dorsolateral striatum, an area crucial for habit formation, perhaps even more than in the nucleus accumbens, which is more about initial reward. This could drive that faster shift from liking the drug to compulsively wanting it.
A shift towards habit.
Potentially. Plus, there are baseline differences, too. Males have the SRY gene on the Y chromosome, which can influence dopamine synthesis. Women tend to have lower activity of the COMT enzyme in the prefrontal cortex, which breaks down dopamine. So their baseline dopamine tone might be different to start with.
So many layers. What about ADHD, attention deficit hyperactivity disorder? We hear a lot about boys being diagnosed more often.
We do. Boys are diagnosed maybe 2 to 4 times more often than girls in childhood, but that's largely because they more often display the classic externalizing symptoms, the hyperactivity, the impulsivity, that are easier to spot and more disruptive in a classroom setting.
Right, the stereotypical bouncing off the walls image.
Exactly. Girls with ADHD, on the other hand, more frequently present with the inattentive subtype. They might be seen as daydreamy, disorganized, easily distracted, but not necessarily disruptive.
So their symptoms get missed more easily.
Very often, yes. Which is likely why the diagnostic ratio becomes much closer to 1.1 in adulthood, as more women are finally diagnosed later when their coping strategies perhaps aren't enough anymore.
Is there a dopamine-related developmental reason for the difference in childhood presentation?
There's a fascinating parallel in animal models. Male rats show this massive overproduction of D2 receptors in the striatum during their adolescence, which is then followed by synaptic pruning, a reduction. This developmental trajectory mirrors the peak of hyperactive symptoms in many human boys, which often lessen after puberty.
And females.
Female rats show a much more modest, gradual change in their D2 receptor density during puberty, less dramatic fluctuation.
Interesting. And does estrogen play a role in ADHD symptoms in women?
It certainly seems to. Many women with ADHD report that their symptoms, particularly executive function difficulties and inattention, get noticeably worse during low estrogen phases of their menstrual cycle or after menopause. This fits perfectly with what we know about estrogen generally enhancing dopamine transmission, especially in those prefrontal circuits vital for attention and control.
So when estrogen dips, symptoms can flare up.
That appears to be the case for many women.
Okay, one more major area. Major depressive disorder, MDD, and specifically anhedonia, that inability to feel pleasure, which is so tied to dopamine. MDD is twice as common in women. How does sex differences fit in here?
The Stark 2.1 prevalence ratio for MDD in women is a huge indicator of sex-specific vulnerability. Anhedonia, that loss of pleasure or interest, is a core symptom, and it's strongly linked to dysfunction in the dopamine reward system.
Right.
And we're seeing sex-specific patterns here, too. Resting state fMRI studies show different patterns of neural activity related to anhedonia severity in men versus women, particularly in key nodes of the reward and salience networks.
So the brain correlates of anhedonia look different.
They can, yes. There's also emerging evidence about specific receptor complexes. For instance, females seem to express a higher density of unique signaling units called D1D2 receptor heteromers in the striatum.
Heteromers.
Yeah.
Like two different receptors sticking together.
Exactly, forming a unique functional unit. It's hypothesized that having more of these specific complexes might, in some way, increase female predisposition towards depressive-like or anxiety-like states, perhaps by altering dopamine signaling dynamics, but that's still an active area of research.
This has been incredibly insightful. I mean, the overwhelming conclusion, it feels irreversible really, is that the human dopaminergic system is fundamentally sexually dimorphic. We just have to move past that old sex-neutral model.
Absolutely. The evidence is just too strong. We need this more nuanced framework. Thinking about the amplitude-driven male system built for high-capacity output, greater stimulus-evoked release, and contrasting that with the hormonally dynamic, efficiency-driven female system.
Right, the female system with maybe lower amplitude release in some contexts, but compensating with that enhanced receptor sensitivity and those unique estrogen-dependent pathways.
Precisely. That framework helps make sense of so much of the data.
And recognizing this fundamental difference has massive implications for medicine, right? That one-size-fits-all approach to diagnosing and treating dopamine-related disorders just isn't cutting it. It's demonstrably suboptimal, as the research puts it.
It really is. We've touched on examples. ADHD criteria often failing girls. Women with schizophrenia often needing lower antipsychotic doses or having different side effect profiles. Women with Parkinson's being more prone to LIDs. It's clear we need change.
So the future has to be about precision medicine, incorporating sex as a fundamental variable.
Without a doubt, it needs to be standard practice. This could pave the way for genuinely sex-specific therapies. Imagine targeted hormonal treatments, maybe selective estrogen receptor modulators, SERMs for women with schizophrenia or cognitive issues, or perhaps even adjunctive testosterone for men struggling with severe negative symptoms.
That would be a huge shift. So looking ahead, where do we need to focus the research efforts? What are the biggest gaps?
Well, first, while the animal models are powerful, we need more confirmation in humans using advanced neuroimaging, like sophisticated PET scans that can measure dopamine release and receptor levels more accurately in living people.
Bridging that gap between animal models and human brains.
Exactly. Second, so much research is just a snapshot in time. We desperately need near longitudinal studies. We need to follow individuals across key life stages, puberty, through menstrual cycles, during pregnancy, across the menopausal transition, to really map how these hormonal shifts impact dopamine circuits and vulnerability to disorders over a lifespan.
That makes total sense. And should we also be thinking beyond just a simple male-female binary?
Critically important, yes. Future research has to grapple with the complex interplay between biological sex, gender identity, lived experiences, sociocultural factors. How do all these things interact to shape the dopamine system and mental health? It's not just about chromosomes and hormones in isolation.
A much more holistic view.
Definitely. And finally, the discovery of these, non-canonical sex-specific pathways like that NMDA-independent LTP in females, that's incredibly exciting. It opens up totally new avenues for drug discovery. We could potentially develop drugs that target these female-specific mechanisms, moving beyond treatments that were largely discovered and validated using male animals or male-predominant study populations.
That's the promise of truly personalized medicine. So wrapping up, this isn't just about satisfying scientific curiosity, is it? It's fundamentally about building a more complete, more accurate picture of the human brain for everyone.
Exactly. It's about working towards treatments that are genuinely more effective, more personalized, and ultimately more equitable for the millions, literally millions of men and women affected by disorders linked to the dopamine system.
So for everyone listening, maybe a final thought to take away and ponder. How might recognizing these fundamental sex-based differences in our brain's operating system, how might that reshape not only medicine, but our approach to everyday health, wellness, maybe even our understanding of our own daily experiences? Could it unlock new possibilities for personal optimization for every single one of us?
Concierge Care, providing world-class, personalized care for patients and members, taking a proactive approach by offering its free digital health clinic for anyone in need of these services. Reach out to us via our website, www.health.conciergecare.net, or give us a phone call at 786-809-2345.